The following is an original production of AZPM News.
Arizona Science is supported by Research Corporation for Science Advancement.
For Arizona Public Media, I'm Tim Swindle, Professor Emeritus of Planetary Science at the University of Arizona, and this is Arizona Science. Joining me today is Martha Bhattacharya, an Associate Professor of Neuroscience at the University of Arizona and the speaker next week in the University of Arizona College of Science Lecture Series. Martha will be talking about how scientists use fruit flies to investigate all sorts of human diseases. Welcome, Martha.
Thanks for having me.
What makes fruit flies valuable for the sorts of studies that you do?
Fruit flies have a very similar genome to humans. And so if we want to understand what's happening inside a human body but may not have the access that we need, fruit flies are a nice alternative that have a really quick breeding schedule. So we can actually look at many animals at once and we can get a lot of information. They also have a very complex nervous system. And so to study something that might happen in the human brain, we can actually use fruit flies as a kind of proxy for those types of studies.
You say they have a complex nervous system. Do they have a nervous system that's similar enough to a human nervous system that you can actually deal with things that you want to in the human?
Yes, absolutely. So they have a central nervous system like we do, which is represented by our brain and inside our skull. And then they also have a descending nervous system, which is the motor control, like moving your appendages, your hands and your toes. The fruit flies have the same type of organization, and so we can use the principles that are set up in the fly nervous system to understand how those complex movements also happen in humans.
Are you trying to understand the nervous system in general, or are you going after, for instance, specific diseases more often?
So my lab mostly works on the early stages of neurodegenerative diseases. We're fairly agnostic to which diseases we might apply our work towards because often the questions have application in multiple areas like Alzheimer's disease or ALS, for example. Alzheimer's disease is more cognitive, ALS is more motor, and we think that a lot of the same processes are happening in both.
Do fruit flies actually get these kinds of diseases?
Not naturally, but they do age. And as we age, we often are more susceptible to age-related diseases, which these are some of, the Alzheimer's and ALS. And so we can express human genes in a fruit fly and start to see similar types of changes, degenerative changes in the fruit fly nervous system as you might see in a human nervous system. So we can correlate those changes we see in flies with things that also happen in human cells and know that those are conserved and therefore that feel more confident that the fruit fly is a reasonable model for something that we might want to know about in a human disease.
Do you study the changes by studying the biology of the flies, or is it more the behavior? Do they exhibit similar behaviors?
We actually do both. So we can, for example, express a gene that makes the fly sensitive to developing ALS. And we can see behaviors that make sense if a fly would have ALS. So changes in motor weakness, movement, we can look at them walking around and see that process get worse over time. But then we can also look at the nervous system directly. So we can dissect out the brain or the equivalent of the spinal cord, and we can look at the neurons that control those movements and see if they are dying or if they have changes that would represent degeneration in that system. And then we can maybe try to rescue those things by repairing some of the processes we think are happening. And that's one of the main things that we do in the lab.
A fruit fly is a pretty small animal, and you're looking at just the nervous system of it. Do you have difficulties in getting to that level of detail?
Well, one thing I'll say is that if you want to look at the fruit fly's nervous system, I wouldn't drink too much coffee first thing in the morning because you do have to do a fairly detailed dissection. But once you get it out of the exoskeleton and put it on a slide, you can use all sorts of great microscopy techniques to magnify the things that you would really like to see. So in practice, I actually don't think it's any harder to look at a fruit fly's nervous system as to look at a mouse or a rat.
Now, it's not just fruit flies that you study, though, correct?
That's right.
What else do you work with?
One of the ways that my lab tends to think is that if we start in something simple and then move to something complex, we can address questions that are conserved across evolution. And so we work on mice and also human cell types. I guess the way that we usually do our experiments is we start in the flies. We ask some questions where maybe we can look at a lot of flies at once or a lot of genes at once and come to sort of a subset of the genes that matter. We can then go into mice and ask the same questions, but maybe we do them in a more simple way where we're not doing full animal behavior. We can look at specific types of neurons that may match what we see in humans better in the mouse brain than in the fly brain. Then the human stuff that we do is based on using induced pluripotent stem cells, which we can turn into different types of neurons or muscle cells that we might be able to look at in a human. Are those same changes happening? So we sort of run the spectrum, and it's fun to actually start projects in one place and then take them beyond that and try to make them relevant to the human condition.
How long does it take to go from the stage of understanding these fundamental things in fruit flies to actually mitigating a disease in humans?
That's a really good question, and it takes longer than anyone might like. But I think what we're starting with is asking the basic biology question. And when you do that, you can know that you're working in a space that makes sense for human cells by looking across these different animals, the mice and the humans and the flies. But translating it into, for example, a drug treatment for ALS would take a long time. One nice thing about the fruit flies is that people can do drug screens in intact animals at high throughput because there are so many animals that we can use that we can, for example, screen through 100 compounds by feeding them to the flies and then asking if that modifies a disease behavior like this motor behavior like ALS. You can do that fairly quickly and simply in a fruit fly, not so simple in a mouse, and maybe not quite as relevant in a cell type where you don't have an intact nervous system to study, right? So I think the flies give us some advantages there to try to start to move towards therapies, but it takes a very long time.
You said that you're kind of agnostic about which disease you're attacking. Mm-hmm. Do you start with the fruit flies and say, I'm going to throw a bunch of stuff out and see what happens? Or do you start with a disease and say, oh, I wonder if we could do that in a fruit fly?
My lab starts with questions about how the processes of neurons that connect one neuron to another degenerate in injury and disease. But that process of disconnection and degeneration happens in many different diseases. It happens before cells die. And so it's one of the places where maybe if you interfere or can regrow those connections, you could mitigate the symptoms of a lot of different diseases where you disconnect parts of the nervous system. So when I say I'm agnostic to disorder, I think it comes from the standpoint of working on a process that could have impacts in many different areas, just depending on the place in the brain that we might look or care about for a particular disease.
In other words, the work you're doing is a little more fundamental than a specific disease.
That's right.
What got you started on this kind of work?
I'm really interested in basic science. When I was a graduate student, I worked on a really fundamental question of the sense of touch. How does the sense of touch work? How do mechanical forces cause a neuron to turn on and fire and transmit information? And that kind of got me hooked onto just trying to ask simple questions about the way the nervous system works. I moved to fruit flies later in my career because I realized the advantages that you could have by using these as a platform to identify new genes that could play a role in some process. And so that was a transition and a learning experience for me. But I also realized that in studying this process of axon injury and axon degeneration, that I could ask a basic question that really would have impacts on a lot of different areas. And so we've mentioned, some of the neurological diseases, but these even extend towards things like chemotherapy-induced neuropathy, which happens to a lot of cancer patients when they take treatments like Taxol or other types of chemotherapy drugs. And so I think that I got hooked on asking the basic questions that might have multiple ways of relevance to human conditions. And so that's sort of where this came from. I also like fruit flies because I think they're really simple animals that students really like working with. And so I was able to recruit undergraduates into my lab and graduate students into my lab with the promise that these are quickly generating organisms and you can get data on your question relatively quickly in an intact system. And so a lot of students really gravitated towards that, and that's helped me get my science to find some results the students enjoy really working with those.
This is fascinating work that you're doing, and I'm looking forward to hearing your talk.
Thank you.
This is Tim Swindle, and you have been listening to Arizona Science. Our guest today has been Martha Bhattacharya, a neuroscientist who studies the nervous systems of other animals to understand humans. She will be the speaker in the University of Arizona College of Science lecture series at 7 p.m. next Wednesday at Centennial Hall on the University of Arizona campus. You can hear more of this conversation and others on our Arizona Science segments at azpm.org.
Thank you to Research Corporation for Science Advancement for their support of Arizona Science.
This has been an original production of AZPM News.